the S-adenosylmethionine because it's a CH3 donor. Phenylethanolamine N-methyltransferase causes one methyl group to be donated to the norepinephrine, and it is converted to epinephrine and stored in the adrenal gland. And it's also a non-specific enzyme, and inhibition can decrease adrenal what you call as the norepinephrine regulated by glucocorticoids. Especially this is regulated by glucocorticoid. Whenever there is more and more glucocorticoids are released, then more and more epinephrine is released from the adrenal gland. In the adrenal medulla, so the catecholamines are stored in chromaffin granules. It's known as chromaffin granules, and these vesicles contain approximately high concentration of the catecholamines, approximately 21% of the dry matter, especially the adrenal medulla. If you cut open it and dry, then 21% of it is containing the catecholamine. And apart from this one, the ascorbic acid is present, and the adenosine triphosphate is present, and the chromogranules are present, whereas the enzyme which converts this norepinephrine to epinephrine and also dopa to dopamine is dopamine beta hydroxylase, DBH is present. And apart from this one, enkephalins and neuropeptide Y, these are the contents of the adrenal medulla, whereas the adrenal cortex do contain the corticosteroids, and they are very important for the release of the catecholamines from adrenal medulla. So the entire combination of the adrenal medulla is 21% is containing the catecholamines, the ascorbic acid, remaining is ascorbic acid, ATP, chromogranules, and DBH, dopamine beta hydroxylase, and enkephalins, and also the neuropeptide Y. See, this 5-HT, 5-hydroxytryptamine, can be produced by aromatic L-amino acid decarboxylase or dopa decarboxylase from 5-hydroxy N-tryptophan. So 5-hydroxyl-tryptophan is just like the tyrosine, and it's uptaken into the exoplasmic membranes. Then it is converted to 5-hydroxytryptamine. And dopamine beta hydroxylase also converts dopa to dopamine, and methyl dopa is converted to alpha methyl dopa, which in turn is converted by dopamine beta hydroxylase to a false transmitter, because it's a what you call as a substrate for the dopamine beta hydroxylase enzyme, and the false transmitter is alpha methyl norepinephrine. Once again, another methyl group will be added to the norepinephrine, and it's called as the what you call as the alpha methyl dopamine, and dopamine beta hydroxylase is the enzyme which is involved, and this is a false transmitter theory or false transmitter. The hydroxylation of tyrosine generally is regarded as the rate limiting step. That's the tyrosine to dopa, conversion of tyrosine to dopa, with that it's a rate limiting step in the biosynthesis of catecholamines, and tyrosine hydroxylase is activated following stimulation of the adrenergic nerves or adrenal medulla. So whenever a neurotransmitter to be released, there should be an enzyme which is very specific to that of the particular step, and this tyrosine hydroxylase is a very specific enzyme which converts tyrosine to dopa, and it's activated following the stimulation of adrenergic nerve endings or adrenal medulla by corticosteroids. And this is a very important step here, and you can learn this particular step. Like this is a sympathetic nerve. Here it's a sympathetic nerve, and this is a effector cell. Here this is a effector cell, and the tyrosine is taken up from the periphery into the cytoplasm of the sympathetic nerve, and tyrosine is converted to dopa to dopamine, and dopamine to norepinephrine. It's converted to norepinephrine. So norepinephrine is going to be taken up, especially in the vesicles, vesicular uptake. Then it is going to have the fusion of many vesicles. Then it is going to be released by the arrival of the action potential. Then the released norepinephrine may act on alpha type of the receptor or beta type of the receptor, especially in the neuroeffector junction. And apart from this one, it may also act on the presynaptic alpha receptor and the going to change. Then here you can see that the monoamine oxidase, that's the first enzyme, is going to act on the norepinephrine and going to break down it to a metabolite, inactive metabolite, which is present in the sympathetic nerve varicosity, that is the exoplasm. And these are all the uptake ones. Norepinephrine is what you call as gives a feedback mechanism. If the concentration of norepinephrine is going to be increased, then it gives a feedback mechanism, negative feedback mechanism, so that the further metabolism of tyrosine will not occur. Usually, the norepinephrine is going to be released after exocytosis, and there is a response beta or alpha receptors. Through alpha or beta receptor, there will be a response will occur after the post-junctional activity. So this entire diagram explains the synthesis, the storage, the release of norepinephrine, and also initiation of the post-junction activity, and also the metabolism of the norepinephrine. Here you can see this entire step with different letters. Yes, the enzymes that participate in the formation of norepinephrine are synthesized in the cell bodies of the neurons and are then transported along with their axons to their terminals. So these enzymes, especially dopamine beta hydroxylase and tyrosine hydroxylase, these enzymes are formed in the cell bodies of the neurons and then are transported along the nerve terminals. So in the course of the synthesis, the hydroxylation of tyrosine to dopa and the decarboxylation of dopa to dopamine like takes place in the cytoplasm, or we can name it as the exoplasm. About half of the dopamine formed in the cytoplasm is then actively transported into the dopamine beta hydroxylase containing storage vesicles, where it is converted to norepinephrine. Here you can see the step that dopamine beta hydroxylase, this particular step you can see. Yeah, please remember this particular diagram, which is a very basic or fundamental to all the next processes, because it is very important to remember tyrosine converted to dopa with the help of the enzyme. First enzyme is tyrosine hydroxylase, tyrosine hydroxylase, which causes one hydrogen moiety to be attached to the benzene ring, which is later the dopa decarboxylase or L-amino acid decarboxylase enzyme. It is an enzyme. Then dopa to dopamine is converted, because here this OH is removed and one hydrogen moiety is added. Then dopamine beta hydroxylase is enzyme which converts dopamine to norepinephrine. Dopamine beta hydroxylase, in the beta position one hydroxyl group is added, whereas the norepinephrine is going to be converted to epinephrine with the help of phenylethanolamine N-methyl transferase. This is the sequential structure. So usually the dopamine beta hydroxylase it is converted to norepinephrine. The remainder which escaped captured by the vesicle is deaminated to 3,4-dihydroxy phenyl acetic acid, or it's also known as DOPAC, 3,4-dihydroxy phenyl acetic acid, and subsequently it is methylated in the O position to homovalenic acid, homovalenic acid, or it's also known as HVA. So this is about the metabolism of the norepinephrine. See, the dopamine beta hydroxylase is going to convert norepinephrine to to dopa to norepinephrine, where it is converted to norepinephrine, whereas the remainder escaped captured by the vesicles and it is deaminated by 3,4-dihydroxyphenylacetic acid. It's also known as DOPAC, and subsequently O-methylated to homovalenic acid. This is the metabolism pathway of the norepinephrine. The adrenal medulla has two distinct catecholamine containing cell types. One is with norepinephrine and those with primary epinephrine. So, the adrenal medulla, it's a very good storage house of the norepinephrine and epinephrine, both are there, and it is containing the two cell types, one with norepinephrine and primary epinephrine. The later cell population, especially the primary epinephrine, contains the enzymes phenylethanolamine N-methyl transferase. That's the enzyme which converts norepinephrine to epinephrine. So it is known as phenylethanolamine N-methyl transferase because one methyl group is transferred to norepinephrine and that norepinephrine becomes epinephrine. And in these cells, the norepinephrine formed in the granules leave the structure presumably by diffusion and is methylated in the cytoplasm to cause epinephrine, that you have very specifically seen in the diagrammatic representation. And then what happens? Epinephrine then re-enters the chromatin granules where it is stored until it is released. So epinephrine is stored in the chromatin granules. Chroma means the colored granules, chromatin granules, where it is stored until the release. The release of the epinephrine occurs after the stimulation of adrenal cortex and release of the corticosteroids. That was flight or fight reflex. So epinephrine accounts for approximately 80% of the catecholamines of the adrenal medulla, with norepinephrine making 20%. So the ratio of epinephrine to norepinephrine in adrenal medulla is epinephrine to norepinephrine 80% to 20%, 80 to 20. So that's why the tissue is made always responsive and a massive epinephrine will be released from the storage site and acts on the different cells. So here you can see that three enzymes, these three catechols, DOPA, dopamine and norepinephrine. These are the three catecholamines which are synthesized with the help of varieties of the enzymes and some of the substances. The tyrosine is uptaken into the exoplasm by the tyrosine beta hydroxylase enzyme and it's converted to DOPA, DOPA to dopamine and dopamine to norepinephrine, norepinephrine to epinephrine. So these are stored in the storage vesicles, especially in the presynaptic nerve terminals. So then they are going to be fused and exocytosis occurs and the neurotransmitter acts on the postsynaptic receptors. And you can see that the tyrosine is uptaken from the nerve endings, that's the rate limiting steps, which is acted by tyrosine hydroxylase enzyme. And the synthesis of norepinephrine depends on the size of the store available for release from the adrenal medulla, the level of glucocorticoids secreted by the adrenal cortex. So it's very, very interrelated. Whenever the glucocorticoids are secreted, they are going to act on the cells, especially the these chromatin cells, especially on the chromatin cells, then they are going to be acted on the postsynaptic vesicle. And in adrenal medullary chromaffin cells, synthesis of phenylethanolamine N-methyl transferase, that's the last enzyme which is required to convert norepinephrine to epinephrine. The synthesis of this phenylethanolamine N-methyl transferase takes place. The activities of both tyrosine hydroxylase and dopamine beta hydroxylase increased in the adrenal medulla when secretion of glucocorticoid is secreted, because the glucocorticoids are the one which make lot of epinephrine to be released from adrenal medulla. So the synthesis of both the enzymes required for the synthesis of the norepinephrine and epinephrine, like tyrosine hydroxylase and dopamine beta hydroxylase, they are increased. And secretion of corticotrophin metabolize the hormones of both adrenal cortex. Predominantly the cortisol is present, cortisol and the medulla. The medulla contains the epinephrine, whereas the adrenal cortex contains the cortisol. So the cortisol is released into the bloodstream, then it's going to act on the receptors of the medulla and releases the epinephrine. They are highly interrelated. Here in this particular diagram, you can very clearly see that the tyrosine transporter is present and tyrosine is uptaken into the exoplasmic exoplasm, and then tyrosine is converted to L-dopa or dopa, tyrosine hydroxylase, or the tyrosine hydroxylase is going to convert tyrosine to dopa. Dopa is converted to dopamine with the enzyme dopamine decarboxylase, and dopamine to norepinephrine is by dopamine beta hydroxylase is the enzyme. And so this is uptaken, and the 3,4-dihydroxymandelic acid will be formed after the action of monoamine oxidase on the norepinephrine, whereas the calcium, whenever this type of reactions are occurring, then exocytosis occurs due to the increased calcium content because of the voltage gated calcium channels. Catechol O-methyl transferase and monoamine oxidase are the two important enzymes which are what you call as the destroy the norepinephrine, or especially the particular the destruction of norepinephrine or epinephrine or both. Catechol O-methyl transferase, it is converting norepinephrine to normetanephrine. So the monoamine oxidase, it acts on the norepinephrine and forms 3,4-beta hydroxymandelic acid. Then this is converted to vinyl mandelic acid, or it's also known as VMA. Then they are all interchangeable, especially normetanephrine. Catechol O-methyl transferase acts on the norepinephrine to cause this particular normetanephrine. Normetanephrine is going to be converted to vinyl mandelic acid, and this is going to be excreted through urine. So this entire diagram is a very informative, because here it informs about the synthesis, the storage, then release, and the metabolism of the neurotransmitter norepinephrine or epinephrine, and also the post-junctional activity, because the norepinephrine is going to bind with either alpha or beta type of the receptors. Here the alpha or beta type of the receptors are present, and the metabolism of norepinephrine in the synaptic cleft occurs by catechol O-methyl transferase enzyme, whereas inside the cytoplasm or the exoplasm, monoamine oxidase is the very specific enzyme. So hope you understood this entire concept.